Hello and welcome to the episode 4 of the series Enzymology for Recombinant DNA Technology. Myself, your host, Dr. Deepak Verma. So today we start from phosphatase in this list number 4. Phosphatase, here we discuss about the alkaline phosphatase. So for wasting our time, just go to this. So phosphatase, alkaline phosphatase, the EC number is 3.1.3.1. This is in short form, it is written as A capital A A's are non-specific phosphomonoesters with optimal activity at alkaline pH as opposed to acid phosphatase which are optimally active at low pH. Alkaline phosphatase catalyzes the hydrolysis of wide variety of phosphomonoesters to the corresponding alcohols and inorganic phosphate. This can also act as phosphotransferase in presence of phosphate acceptors such as ethylamine or trace. This alkaline phosphatase is widely distributed in prokaryotes as well as in eukaryotes. All purified enzymes are dimers, zinc metalloenzymes with an active site, serine, which forms a covalent phosphoserine intermediate during catalysis. The molecular ranges from 80 to 220 kilodalton per dimer, and mammalian enzymes are generally larger than the bacterial enzyme. Plants are rich in non-specific acid phosphatase, but not in alkaline phosphatase. Although there are many sources. So commercially, this enzyme is isolated from bacterial and calf intestine, and also humans also. So, eco- I'm going to little discuss about every enzyme. So, for bacterial alkaline phosphatase is isolated from Escherichia coli, which is a non-specific phosphomonoester which catalyzes the hydrolysis of 3' or 5' terminal phosphatase from DNA, RNA, and a wide variety of organic phosphomonoesters. The phosphate reaction products are DNA or RNA with 3' hydroxyl or 5' hydroxyl termini and inorganic phosphate. Now we comes to the directly to the calf thymus, calf intestinal. So this alkaline phosphatase from isolated from mucosa lines of intestine and has many catalytic property in common with bacterial alkaline phosphatase. This is also a dimeric zinc metalloenzyme, but it has a slightly larger molecule size, 130 kilodalton approximately. On one notable distinction in calf intestinal alkaline phosphatase is heat labile compared with the bacterial alkaline phosphatase. This property alone makes this a valuable alternative to bacterial phosphatase in many applications requiring successive treatment with the other enzymes. And for humans, there are at least 4 distinct forms of ATPase: placental and placental like germs, uh, germ cells, intestinal and liver, body, bone, kidney, ATPase. The germ cells actually normally found in trace amount in the testis and the thymus. The human alkaline phosphatase is clinically important makers of various disease and cancers. Like carp intestinal one, all human alkaline phosphatase are dimeric zinc metalloenzymes. They can distinguish by immunological and various physiochemical properties. Those included thermal stability, phosphatidylate, hydrolytic activity, and kinetics of denaturation in presence of guanidine hydrochloride or urea. So now we comes to its biochemistry. So alkaline phosphatase is as very important enzyme for this recombinant DNA technology. So by this substrate specificity, so what are its substrates? So generally for recombinant DNA technology, we use bacterial alkaline phosphatase. So here I am discussing about bacterial one. So first is the phosphomonoester. So bacterial alkaline phosphatase, in short form I can say BAP, is a non-specific phosphomonoester. The 5' and 3' terminal phosphatase of DNA, RNA and oligonucleotides are substrate, and a host of other alkyl and aryl organic phosphates. And next is the substrate termini structures, means that nick and gap. What about that? So at a temperature below 37 degrees centigrade, this BAP quantitatively hydrolyze external phosphomonoester, but not the phosphomonoester located at nick. Means it hydrolyzes only the external phosphomonoesters, not that located in the nick, at the nick. At evaluated at high temperature, approximately this above than 65 degrees centigrade, both types of phosphomonoesters are hydrolyzed. So now phosphoric acid anhydride, simple acid anhydride of orthophosphoric acid, and short chain metaphosphate are hydrolyzed in the same fashion and at the same rate as phosphate monoester. And next is the phosphate ester analog, such as sulfur substituted analogs. These are also its substrate, and phosphoramides, phosphate with C substitution, phosphate with F substitution. These all are the its also this can also hydrolyze this type of substrate. Phosphate acceptor substrates also in as presence of the high concentration of acceptor substrates such as trace 1 molar. Normally we use 1 molar, so ethylamine and glycerol. Bacterial alkaline phosphatase transfer the phosphate group from the donor substrate to hydroxyl group of the acceptor, resulting the transphosphorylation. The alkaline phosphatase can synthesize inorganic phosphate via similar phosphoryl transfer. Now, up to here, I have discussed about this substrate. Now we comes to the application. So the application of bacterial alkaline phosphatase can be divided into the 4 categories: as simple phosphomonoester reagent for dephosphorylating terminal phosphatase phosphate of nitric acid number, and then next is the as detection tool in oligonucleotide or protein conjugated systems, and next is the as a reporter enzyme in the expression of a alkaline phosphatase fusion gene, and the last fourth is the as the leader in extracellular secretion of fusion proteins. So these are the application for this alkaline phosphatase. Now we move to the kinase enzymes. Polynucleotide kinase, its polynucleotide 5' hydroxyl kinase. This EC number is 2.7.1.78. So kinase actually are extremely diverse group of enzymes that catalyze the transfer of gamma phosphoryl group from NTP to the acceptor molecules. NTP means any nucleotide, any kind of nucleotide. Depending on the nature of phosphate acceptor, kinases are largely divided into the protein kinase, carbohydrate kinase, and polynucleotide kinase. Kinases are uniquely present in the nature and in various tissues and organisms. They play essential roles in cellular metabolism, regulation, and replication. So here we will discuss about polynucleotide kinase, especially from the phage, bacteriophage T4. This bacteriophage T4 polynucleotide kinase is perhaps the best characterized among all known kinases from the various sources such as rat, liver, calf thymus, etc. etc. In general, this polynucleotide kinase has two types of activities. One is phosphorylation and another is 5' phosphate exchange reaction. So here I discuss about its substrate specificity is on phosphorylation. Before discuss about that, this is its international unit definition that states that one unit is usually defined as the amount of enzyme which catalyze the transfer of 1 nanomole of PI in 30 minutes at 37 degrees centigrade. Sorry. So now I come to the substrate specificity. The kinase reaction catalyzed by T4 polynucleotide kinase is reversible. For the single strand DT, the substrate at pH 8, the equilibrium has been estimated to be 50-fold in favor of the forward reaction. So, for practically, we use therefore each direction for this catalyzed reaction will be described if they are irreversible. Anyway, so what are the substrates? Donor substrate. As phosphate donors, CTP, GTP, UTP are equally effective as ATP with same Km and all these things, also approximately 20 micromolar. In reality, DATP also functions as effective donor substrate for this. Now we come to the acceptor substrate. All 5' hydroxyl of DNA, RNA, or oligonucleotides are substrate for phosphorylation. And terminal structure of DNA preferred substrate for single strand oligonucleotide or double strand oligonucleotide with protruding 5' hydroxy terminal. With these substrates, a rapid, essential, complete phosphorylation is achieved at an ATP concentration of almost 1 micromolar. And this ATP concentration is an 2 to 5 fold excess over the concentration of the 5' hydroxy terminal. And preference for terminal nucleotides, this T4 polynucleotide kinase displays some degree of preference towards the 5' terminal nucleotide at a constant ATP concentration. For homo decanucleotide varies from 15-fold, and modified terminal nucleotides such as methylated G residues, A purine sites, and nucleotide with modified sugar. These all are the substrate for this bacteriophage T4 polynucleotide kinase. And sometimes non-nucleotide substrate is also such as aromatic hydrocarbon trials and benzopyrene also substrate for this polynucleotide kinase, whereas diols, phenols, triols, and parental hydrocarbons are not for its substrate. Now we move to its applications. So phosphorylation actually the reversible nature of T4 polynucleotide kinase catalyzed reactions and distinct 3' phosphatase activity render the PNK extremely useful in a wide variety of applications. The reaction condition can be greatly adjusted for respective 5' phosphorylation, dephosphorylation, and phosphate exchange reactions. Phosphorylation number one is the application phosphorylation or radio labeling of 5' hydroxyl of DNA and RNA. The 5' labeling of nucleic acid by radioactive phosphorus or using this T4 PNK is often a key step in the number of important techniques in molecular biology, including chemical, enzymatic, and dideoxynucleotide sequencing of DNA and RNA. Next is detection and assay of endonuclease activity. This T4 PNK catalyzed 5' phosphate exchange with radioactive phosphorus in DNA fragments is sensitive and reproducible method to assay nuclease activity. And next is a radioactive phosphorus post-labeling assay. These assays permits detection of femtomole level, means 10 to the power minus 12 level of base and sugar lesion of DNA caused by the various exogenous factors, including radiation from X-ray, UV, nucleation, and other chemical addition. And next application is the use of 3' phosphate activity. T4 PNK can be used as a specific 3' phosphatase. It has been employed in the studies of DNA damage and repair to determine the presence of 3' phosphate terminal on the radio-labeled 5' end-labeled DNA fragments. The removal of the 3' phosphate is accompanied by the decrease in mobility of such fragments on sequencing gels equivalent to 0.5 to 1 nucleotide position. And the last application is also a specific application that lacking the 3' phosphatase activity. This mutant enzyme is uniquely suitable for the preparation from 3' NMP of 3.5' biphosphate, which can be used for synthesis and labeling of DNA and RNA. Now we come to our next enzyme, that is DNA polymerase. And as we know that these are the group of polymerase that catalyze the synthesis of polyribonucleotide from monodeoxyribonucleotide triphosphatase, performing the most fundamental functions in vivo of DNA replication, repair, and in some cases cell differentiation. In fact, different types of DNA polymerase have been found in single organisms. For example, 3 DNA pol 1, 2, and pol 3 in E. coli, or 5 DNA pol alpha, beta, gamma, delta, and eta in higher eukaryotes, which are believed to perform a specialized in vivo functions. With somewhat different complexity from in vivo functions, DNA polymerase can when given a suitable condition, also perform DNA synthesis in vitro. They require an addition of DNTPs and initiating oligonucleotide called primer carrying 3' and hydroxyl group that can be used as a starting point of the chain growth. DNA polymerase cannot initiate synthesis de novo from the mononucleotides. So there are actually a lot of polymerases, but here we discuss about E. coli, T4, T7, and most important TAG1, tag polymerase. So one by one. So first I discuss about E. coli polymerase. I will give you just brief introduction here. E. coli DNA polymerase 1 is a monomeric of about 109 kilodalton multifunctional enzyme that has three major activities: 5' to 3' DNA polymerase, 3' to 5' exonuclease, and 5' nucleases. Pol1 is also known as Corenberg enzyme, is probably the best characterized of all polymerase and serve as prime model for DNA polymerases in the elucidation of polymerase structure and associated multifunctions. Under the condition of limited digestion with substratum, Pol1 is cleaved into the two fragments. The large 68 kilodalton C-terminal fragment process the DNA polymerase and 3' to 5' exonuclease activity. The small 34 kilodalton N-terminal fragment containing 5' nuclease activity. These all the specificity, these all the actually characters we will discuss later in this video. First, the later fragment, actually this means 34 kilodalton, the smaller one, which is better known as kill now fragment, can be replaced in vitro most of the functions of Pol1. In fact, the lack of 5' nuclease activity renders this kill now fragment more suitable than Pol1 in many applications. And for recombinant DNA technology, most of the experiments done by this E. coli polymerase 1. Now we come to the T4 DNA polymerase. The DNA polymerase from the bacteriophage T4 is multifunctional monomeric enzyme of 104 kilodalton, which causes both DNA-directed DNA polymerase and 3' to 5' exonuclease activity, but not 5' nuclease activity. The 3' to 5' exonuclease activity of T4 DNA polymerase hydrolyze both single-strand DNA and double-strand DNA in the absence of DNTP. Compared with the E. coli DNA Pol1, which is functionally a repair enzyme in vivo, the polymerase activity of T4 DNA poly, which is a replicative polymerase, is approximately 2 times higher than, and the 3 to 5' exonuclease activity is 2 order of magnitude stronger. These properties make T4 DNA poly extremely valuable tool in variety of applications, including DNA strand synthesis, manipulating, cloning, and nucleotide sequencing. And now we come to the T7 DNA polymerase. This is a heterodimer of 92 kilodalton enzyme composed of a phage G5 protein and the post-encoded thyrodoxin in a 1:1 stoichiometry. In addition to the highly processive polymerase activity, the native T7 DNA pol has a strong 3' to 5' exonuclease activity specific for both single-strand DNA and double-strand DNA. No 5' nuclease activity has been found with T7 DNA poly. Modified forms of T7 DNA poly form which 3' to 5' exonuclease activity has been removed are commercially available. Now we come to the tag polymerase. The DNA polymerase from Thermus equator is a monomeric enzyme of 94 kilodalton with DNA polymerase and a strong 5' nuclease activity but no 3' to 5' exonuclease activity. Tag poly is a highly thermostable and processive and has a high rate of polymerization. These properties, together with the ability of the polymerase from clone source, make a key enzyme in many applications involving high temperature automated polymerase chain reaction. So now we comes to its unit definition and its substrate specificity. So international unit definition of DNA polymerase is this one unit is the amount of enzyme that incorporates 10 nanomoles of DNTPs into the acid precipitate fill material in 30 minutes. And here for substrate specificity, I will discuss here about only about E. coli polymerase 1 because most of the reactions are done by this only enzyme. So there are three activities I told you earlier. So one by one. So first substrate specificity for polymerase. So template structure. So this enzyme catalyzes template dependent directed polymerization of DNTP on a wide variety of natural and synthetic DNA and RNA. Depending on the structure of the template, polymerization reasons can be discussed later in another video. Now the and primers. So primers actually primers length and mismatching primers is also a thing or that. So with the homopolymeric DNA templates, mononucleotides both DNMP and DNTPs are as well as oligodeoxynucleotide serve as primers of DNA synthesis by polymerase enzyme. Now the nucleotide substrate. So as I told you, deoxynucleotides and nucleotide analogs are its substrate for its substrate. Now the frequency of misincorporation. So the frequency of mismatching nucleotide incorporation by polymerase has been estimated to be very rare, in approximately 1.5 into 10 to the power minus 6. So in the presence of manganese or in place of magnesium ion, polymerase display a substantial degree of specificity relaxation, permitting relatively efficient incorporation of repaired mismatching DNTPs. And polymerase processivity. The processivity of Pol1 ranges between 18 to 15 nucleotide at 37 degrees centigrade at low salt concentration. At the rate of polymerization, the maximum rate of polymerization by E. coli polymerase enzyme on some homopolymeric templates is 350 minutes per minute, although the initial rate can be as high as 600 per minute. And now we come to its another activity, which is 3' to 5' exonuclease activity. So what is the substrate here? So for 3' to 5' exonuclease activity, polymerase requires free 3' hydroxyl terminal in single strand DNA or double strand substrate. The 3' phosphate terminated DNTP is not hydrolyzed by this activity. product profile. The products of 3' to 5' exonucleotide action on single strand DNA or double strand DNA are exclusively NMPs. The rate of exonuclease activity is here turnover number for is 16 to 19 per minute. Now another specificity 5' nuclease activity. Substrate are here are present in that actually this substrate require double strand DNA template with free 5' and nothing else. And the product profile, the products of 5' nuclease catalyzes exhaustive hydrolysis are predominantly 5' mononucleotides and some oligonucleotides. Substrate for endonuclease activity. The 5' nuclease activity is presumed to excite the 5' single strand arm at a junction with the duplex, most likely between the first two base pairs of the duplex region. The 5' arm can be fried or displaced oligonucleotides and separate activity of 5' nuclease domain. The 5' nuclease activity is retained in all small N-terminal fragment produced by limited proteolytic cleavage of polymerase. Now we move to our discussion about another enzyme, RNA-directed DNA polymerase. In simple terms, this enzyme is known as reverse transcriptase. And this is a group of DNA polymerase that catalyzes the synthesis of complementary DNA, the cDNA, from the RNA template efficiently, thus forming reverse transcription. Reverse transcriptase was first found in purified virions of murine like leukemia virus MLV by Baltimore and Brown sarcoma virus by Temin and Nidgen. This enzyme is proteolytically derived from the precursor encoded by the viral Pol gene. Reverse transcriptase uses viral RNA as the natural template to synthesize double-strand pro-viral DNA in vivo, thereby playing a central role during the life cycle of retroviruses. Reverse transcriptase and phenomena of reverse transcription have since been discovered in many other organisms, including DNA viruses, such as hepatitis B virus, Calimero virus, and bacterial in Myxococcus, and some clinical strains of E. coli B, yeast, fungi, invertebrates, such as Drosophila, and plants also. So here we will discuss about AMP transcriptase and MOLB reverse transcriptase. So there are two types of reverse transcriptase used in recombinant DNA technology. AMP reverse transcriptase and the another one is MOL1. So AMP reverse transcriptase isolated from avian myeloblastosis viruses, and it is a prototype of avian retrovirus reverse transcriptase. This enzyme is a dimer of 155 kilodalton and composed of non-identical subunits of alpha and beta. AMP reverse transcriptase has multiple enzymatic activity, including RNA-directed DNA polymerase, RNase H, and DNA endonuclease. AMP reverse transcriptase does not have 3' to 5' or 5' to 3' exonuclease activity. This enzyme is a key reagent in cDNA synthesis and nucleotide sequencing, although its role is now partially shared with cloned M, MOLB reverse transcriptase. And another one is the MOLB reverse transcriptase is mononuclear leukemia viruses isolated from causes both DNA polymerase and RNase H and D activity. It is a monomeric 80 kilodalton in solution, but it is presumed to be function as a dimer. The ratio of polymerase to RNase H activity and many of the MOLV reverse transcriptase functions are similar to those of AMV reverse transcriptase. In contrast to AMV reverse transcriptase, however, this MOLV reverse transcriptase does not possess DNA endonuclease activity. The genes for MOLV reverse transcriptase has been cloned and expressed in E. coli, and both wild-type reverse transcriptase and modified reverse transcriptase are commercially available. MOLB reverse transcriptase is thus reliable reagent which shared with AMV reverse transcriptase much of the role of cDNA synthesis in molecular cloning. Modified MOLB reverse transcriptase has proven to be particularly useful in the synthesis of full-length cDNA. Now we come to its international unit definition and substrate specificity. Here we will discuss about this AMV one. So unit definition is written here. So now we directly come to the substrate specificity. First we will discuss about its polymerase activity. First is template. So homopolymeric and native RNA templates is the preferred substrate. Sometimes it's used also DNA as a substrate. Now the primer and or native primer, synthetic primer, or mismatching primer, or primer of the secondary strand DNA synthesis in vitro, or these are the primers we have used for reverse transcriptase. Now the nucleotide substrate and analogs. Like all DNA polymerase DNTPs are the strand nucleotide substrate for reverse transcriptase. This AMV reverse transcriptase can also be efficiently incorporated in a variety of nucleotide analogs such as DNTPs, alpha S, DDNTPs, and DITPs, and many extra. Now the processivity. The polymerase processivity of reverse transcriptase on the RNA template with oligodeoxyribonucleotide primers is 97 nucleotides. And frequency of misincorporation has very high rate under certain conditions, exceeding more than 10 times of than Escherichia coli DNA polymerase when transcribing homopolymeric RNA, DNA or single strand fast DNA template. And pyrophosphatase exchange and pyrophosphorylases. This reverse transcriptase has no, as I told you earlier, that has no 3' to 5' or 5' to 3' exonuclease activity with high concentration of inorganic phosphatase. However, this reverse transcriptase catalyzes the pyrophosphorylation and inorganic exchange. Now we come to its another specificity, that the ribonuclease, that is RNase H. So first is the substrate and product profile. So in this AMV reverse RNase H degrades the RNA strand of RNA DNA hybrid in both the 3' to 5' and 5' to 3' direction at the same rate. The digestion products are oligonucleotides which ranges its size from the 2 to 30 nucleotides with an average of 10 to 12 nucleotides. Next is the RNase endonuclease activity. This uh, this RNase H is essentially an endonuclease. It can cleave relaxed circular covalently closed plasmid in which 770 consecutive resident of one strand are ribonucleotides. And next is the cleavage of RNA DNA junction. This RNA can cleave in vitro the junction between RNA and DNA, an action suited for its in vivo role in removing RNA primers. Now the processivity of transcriptase. This RNA H activity of AMP reverse transcriptase alpha beta complex is partially processive in vitro, whereas the model of RNase H action of the alpha subunit enzyme is distributive. Now another subject is to say that DNA is endonucleated. So first is the divalent cation requirement. So divalent cation is need of magnesium, and virion purified AMP reverse transcriptase causes a physically associated DNA endonuclease activity. Under certain assay conditions, the endonucleatic activity is hardly detectable. Effect of the, it's the preferred substrate. Preferred supercoiled double-strand DNA is the preferred substrate for this. Now the sequence preference. In the presence of magnesium ion, the DNA endonuclease in the form of pyrolytic fragment introduce a single nick on supercoiled DNA regardless of the ratio of the enzyme of DNA. With the supercoiled DNA containing LTR circles junctions sequence of PP32 enzyme nicks the DNA at one or other of the two sides. And next is the last is the DNA binding activity. This reverse transcriptase demonstrates a strong double-strand DNA binding activity strong enough to retain the enzymes on the NC filter with bound called E1 DNA. This double-strand DNA binding activity is presumed to be a prerequisite to the reverse transcriptase associated endonuclease and the unbinding activity. The endonuclease activity is responsible for viral integration into the host chromosomal DNA. So now we come to the reverse transcriptase application part. So here first we discuss about the synthesis of first and cDNA. Reverse transcriptase plays a key role in the synthesis of cDNA from the RNA templates. The primordial step in the cDNA cloning. When the cDNA are labeled during synthesis, they can be used as hybridization proof as well. So it is used as a conventional first strand of cDNA synthesis. It can be achieved in a number of ways depending on the cloning strategy. And nucleic acid amplification is an isothermal amplification strategy called NASBA. This is actually alternative step of DNA synthesis from RNA template, and RNA synthesis from the DNA template are performed using this reverse transcriptase and T7 RNA polymerase respectively. And this reverse transcriptase is also used as this nucleotide sequencing. Both AMP and MOLB compound named DNA dependent DNA dependent DNA polymerase in the dideoxynucleotide sequencing of DNA, especially at the region of high GC content or secondary structure. Reverse transcriptase has been shown to be particularly useful in the sequencing with fluorescent tag modified ddNTPs that are not subject for polymerase and site-specific forced misincorporation interferences. two sites specific mutagenesis methods have been developed that reverse transcriptase as error-prone polymerase. One is the forced misincorporation mutagenesis, and another is the method of simply extending the primers with mispaired terminal. So, so up to here, this reverse transcriptase is also finished. Now we move another enzyme, the terminal transferase. And generally, it is termed as template independent DNA polymerase. This is a actually group of DNA polymerase which, unlike DNA or RNA directed polymerase, requires neither DNA or nor RNA templates but catalyzed linear consideration linear condensation polymerization reaction. Terminal deoxynucleotide transferase is traditionally written as TDT. This enzyme catalyzes in vitro the polymerization of DNTPs into the 3' hydroxyl group of initiator, oligo, or polynucleotides. So this is its EC number 2.7.7.31. Now we comes to its unit definition, international unit definition, and substrate specificity. Substrate specificity, the first is the initiator. The 3' hydroxyl termini of oligo or polydeoxyribonucleotides serve as initiator of polymerization. The minimum length of initiator substrate is 3-phosphate residues. Trinucleotides with 5' phosphate is also suitable initiator, but trinucleosides diphosphatase are not. And 3' hydroxy terminal, its structural and effect of ions, base sequence preference, unusual initiator substrate is also it's having some factor about its activity. So next is the unusual initiator substrate. So in the presence of various alcohol under conditions resemble the standard polymerization reaction, the enzyme, this polymer enzyme TDT is catalyzes a novel reaction in which phosphodiesters are synthesized and inorganic phosphate is released. With glycerol and DATP as substrates, the product has been identified as 2,3-dihydroxypropyl ether of deoxy adenosine 5 monophosphate. So nucleotide substrate next is nucleotides are DNTPs and ribonucleotides, deoxynucleotides, and some modified nucleotides is also its substrate for activity. And now the tail length. So synthesis of poly AT and poly DT with chain length up to the 20,000 nucleotides proceed reasonably well as done as synthesis of poly C with chain length of up to the 3,000 nucleotides. In contrast to an addition of a DG is self-limiting with only about 20 to 30 nucleotides. So we come to its application. So as the last I discussed the tail length. So homopolymeric tailing is one of its application in which by this enzyme was first and one of the most important technique used in molecular cloning. Regarding the cloning strategy, which involves homopolymeric tailing, many variations exist either in the synthesis of cDNA or in the annealing of ligation of cDNA with the cloning vector or both. So next is the 3' end labeling. TDTase is extremely useful for labeling DNA at the 3' end, either with the ribo radionucleotides or non-radionucleotides. So it's a molecular hybridization probes, special application for nucleotide sequencing and artifact resolution in nucleotide sequencing. This 3' end is used for this purpose. The last application is misincorporation mutations. Because this enzyme catalyzes a linear polymerization of DNTPs without template direction. A mismatch in oligonucleotide can be added to the 3' termini of DNA, especially in the presence of cobalt ion. The misincorporation can be made fairly site-specific. Even the single nucleotide level 3' hydroxyl termini are exposed to controlled digestion with either exo 3 or the other restriction enzyme. Now we move to our last enzyme, that is RNA polymerase. It's a DNA-directed RNA polymerase. So this is actually key enzyme in the transfer of genetic information from DNA to RNA. During this process of information transfer, this is called the transcription, and RNA chain messenger transfer or ribosomal is copied by RNA polymerase from double-strand DNA template. The RNA polymerase elongates RNA chains by forming 3' to 5' phosphodiester linkage using ribonucleoside 5-triphosphate as substrate and releasing pyrophosphatase as reaction product. The mRNA transcripts are subsequently translated into the proteins in the ribosomes called the process called the translation. So here we discuss about three main RNA polymerase: T7 RNA polymerase, SP6 RNA polymerase, and T3 RNA polymerase. So little bit about everyone. I have to going to uh, discuss about little things. So first we start with the T7. This is a single 100 kilodalton polypeptide consisting of 883 amino acid chain length. Despite of its structural simplicity compared with the multimeric prokaryotic and eukaryotic RNA polymerase, T7 RNA polymerase performs a highly specific promoter recognition and all the other steps in the transcription process in the absence of any additional protein factors. And for SP6 RNA polymerase, the RNA polymerase, this is from bacteriophage like other phages T7 and T3 RNA polymerase is extremely useful enzyme for in vitro transcription. This SP6 RNA polymerase possesses a stringent promoter specificity similar to but not distinct from the T7 and T3, making it possible to transcribe bidirectionally in vitro a gene or DNA clone in double promoter plasmid vector. And for T3, this RNA polymerase is from the bacteriophage. T3 is structurally and functionally very similar to T7, yet the T3 RNA polymerase exhibits a distinct nearly exclusive promoter specificity. The consequences of promoter sequence derived from T3 promoter sites on the T3 genome is a 23 base pair conjugate sequence. So we will here discuss about that T7 RNA polymerase, its international unit definition, and now we directly come to the substrate specificity, the promoter characteristic. The promoter recognized by the T7 RNA polymerase can be represented by a highly conserved sequence of 23 conjugates base pair, which includes the site of transcription initiation and extend from the minus the transcription initiation, that is plus one, and the started from minus 17 to plus 6. So the minus 17 to plus 6 promoter defined as the minimum length having the full promoter length. Now the templates. So natural or modified DNA, synthetic templates, and template structure, and also the RNA templates also. These are all RNA templates. Transcript profile. So distribution of the products. So it is very distributedly, and products under the non-standard reaction conditions. Nucleotide 5 just like 5' end nucleotides and 2' deoxyribonucleotides, 3' deoxynucleotide chain terminators, alpha thiophosphate nucleotides. These are the all nucleotides. Even also some other nucleotides, UTP, like they are also used. And processability. The transcription by the T7 RNA polymerase is rapid and processive. Transcription for T7 promoter containing plasmid DNA results in transcript several times in the plasmid length. And the transcriptional terminators. The typical termination signal for fast T7 DNA encoded similarly to the independent terminator of E. coli RNA called the RNA sequence that can form a stable G-series stem loop followed by one of 6 U residues. The application for this T7 RNA polymerase, which is considered to be the representative of fast RNA polymerase, is an invaluable tool used to direct a high-level expression of cloned gene in vivo under the control of T7 polymers promoter, and prepare milligram quantities of almost any RNA transcript in vitro. A number of plasmid vectors that feature built-in single or double phase promoter are available for specialized as well as general gene cloning and expression. These transcription vectors make it much easier and versatile to use fast RNA polymerase in a wide variety of recombinant DNA techniques in conjugation with gene cloning. In vivo use of such fast RNA polymerase promoter expression system provides a mean of gene therapy, vaccination, and phenotypic filtration. In vitro transcribed RNA can be used as diverse applications, for example, as hybridization probe in northern or the southern blots, as substrate for RNA processing and structure function studies, as substrate in exon-intron mapping of genomic DNA, as template for reverse transcription, dideoxynucleotide sequencing, as template for in vitro translation, as capture reagent for RNA binding proteins, and as antisense probe and ribosomes. Some of the features that make the T7 RNA polymerase particularly useful for such applications include a stringent promoter selectivity, a high rate of synthesis, and an ability to produce complete transcript from the almost any DNA that is linked to T7 promoter. Far, yes, T3, T7, and sp6 promoter are not recognized by the bacterial RNA polymerase, but they can be recognized by some mammalian nuclear factors and be transcribed in mammalian cells even in the absence of fast RNA polymerase. Among various transcription systems containing T7, T3, and sp6 or bacterial synthetic consequence promoter, the T7 system is superior to other systems in several respects. So here we are following some examples. So basic principle underlying why application of fast RNA polymerase and conjugate motor system, just like high level of expression of fusion gene. So a relatively small amount of T7 RNA polymerase originating from the clone copy of the Pol gene is usually sufficient to direct a high level of transcription from a T7 promoter in a multi-copy plasmid in E. coli. Both the rate of synthesis and the accumulation of RNA directed by T7 RNA pol can be reached level compared to the dose of rRNA in the normal cells. So in coupling expression system and conjugate expression system and expression system provisions with high level translation all are used in this expression or with the fusion gene. So preparation of labeled or unlabeled RNA transcript in vitro. In vitro transcription of is an important technique for preparing of RNA that can be used for various purposes. At the present, there are no efficient simple techniques available for the synthesis of RNA oligonucleotide comparable to the those of the current automated DNA synthesis. In vitro, transcription is also basis of efficient isothermal gene amplification techniques. In vitro, transcription can be performed in one or the two format by the vector-based approach in which clone target DNA is abated to the T-cell promoter in a linearized polymer, linearized plasmid, or by the synthetic oligonucleotide based approaches in which template DNA is linked to the mobile promoter. Then last is RNA polymerase based RNA sequencing. So RNA sequencing can be directly performed by the double-strand DNA templates cloned downstream of a fast promoter T7T3 species. Transcription reactions are performed in the presence of a chain terminator in a way similar to the dideoxynucleotide sequencing of DNA. The difference is that DNA sequencing by RNA transcription obviates the need to prepare single-strand DNA templates or to use sequencing primers. So these are some applications. Actually, RNA polymerase have much more applications. So I have to make one more video or I have to take one more class for this. Anyway, up to here, all the enzymes for recombinant DNA technology is finished. So thank you for listening to this. Thank you all. Thank you.